Good morning, everybody. My name is Samuele Lamon, and I'm part of the Wild Peanut Lab at the University of Georgia. And one of the things that I really like about being plum breeder and working with crops is all this diversity, all these different shapes, colors, and size. And this genetic diversity is also present in a peanut. Here, for example, you can see peanuts with different kernel colors. However, when we compare genetic diversity of peanut with other crops, we will notice that is greatly reduced. In fact, we say that peanut has a narrow genetic base. And this is due to the peculiar origin of peanut. Peanut is an allotetraploid species with an AABB genome composition, and it was derived from the crossing of two wild diploid Arachis species and followed by genome duplication. And this polyploidization event created a genetic bottleneck. And so the genetic diversity that was present in the wild diploid species was not transferred to our modern peanut. And this is a big issue because when we are dealing with these diseases, peanut has just few traits to fight back, but the traits that will confer complete resistance are present in the diploids. So we have to integrate some diploid background in peanut, and this is a complex process. Fortunately, peanut has some ways to increase its own genetic diversity, and one of these is double reduction. Is a process that was never identified before. So what is double reduction? Double reduction is a genetic process discovered by Darlington in 1929 when he was working with the genus Tradescentia, and he defined where both the chromatids of one chromosome pass in the same nucleus at the second telophase, which means for peanut that if we have a cell with alleles in a single dosage, at the end of meiosis, we will notice that we have gametes in a double dosage for a specific allele. And in peanut in particular, you will notice that we will have chromosome just from one of the two subgenomes. That is something that we will never expect. We will expect to have a chromosome from the A subgenome and a chromosome from the B subgenome. And so when we have one of these gamete, outcome of double reduction, and we cross it with our regular Arachis hypogaea gamete, like in this case with one chromosome from the A subgenome and one chromosome from the B subgenome, we will have unbalanced genomic composition. In fact, you can notice here that the genome is composed by a AAAB composition instead of an AABB composition. And this creates a lot of diversity and novel allelic combinations. And so it creates traits that can be used by plant breeders for their breeding programs and create new varieties. So the hypothesis of this project is that increased genetic diversity is originating from a spontaneous occurrence of double reduction events. And our objective is to estimate the percentage of double reduction in segmental allotetraploid peanut. So with this, the outline of my presentation is to talk about tetrasomic recombination, which is an essential concept to understand double reduction. And then I will talk about how we estimated double reduction in peanut, and I will end with a summary of the results and the future objective of these projects. And so tetrasomic recombination, what is tetrasomic recombination? Tetrasomic recombination is something that happens during prophase first of meiosis. This phase, prophase first, is that phase where the chromosome condense and we have the pairing of the homologous chromosome and the recombination of the genetic material. So in polyploid species, there are two main models during this phase for recombination. The first one is the disomic model, which is typical of allopolyploid species like soybean, and is typical of allopolyploid species because these are species that originated from the cross of 2 or more species, and so they have more than one genome. So in this case, you can see there are 2 subgenomes, and so you can see there is disomic pairing, bivalent pairing, and disomic recombination just within the subgenome. The other model is the tetrasomic model, which is typical of autopolyploid species like potato. And this is typical of autopolyploid because they originated from a single species or a single individual. And so each set of the chromosome is really similar to each other. And so they can pair during prophase first and create multivalents, and their recombination can happen among all the different set of chromosomes. So what about peanut? Peanut is a really peculiar crop. It follows a third genetic model called segmental allopolyploid, which means that it mainly behaves as a allopolyploid species with bivalence and disomic recombination, but there is still a significant level of multivalence and tetrasomic recombination. So why tetrasomic recombination is important to understand double reduction? It is important because 2 out of 3 of the essential events for double reduction occurrence can be simply summarized as tetrasomic recombination. And those are multivalent pairing during prophase first and crossing over between non-sister chromatids during prophase first. The third event is a reductional separation pattern throughout meiosis first and meiosis second. I will explain this concept in a few slides. So here, what you're looking at is a comparison between a regular peanut cell and a cell, which is a candidate cell for double reduction occurrence at prophase first. So here you can see that the chromosome from the A subgenome, they form bivalence, and the recombination can happen just within the specific subgenome. Here you can see that the cell creates multivalence, and the recombination can happen among all the different subgenome. And so we satisfy the first 2 conditions for double reduction to occur. And so at this point, this cell at prophase first needs to go through the first round of meiosis. And so it needs to go from prophase first to telophase first. At this point, this cell can follow 2 different separation models. One is called equational and the other is called reductional. I will talk about just the reductional to keep the things simple for you to understand. So reductional separation simply means that if we are considering a specific allele, let's say the A1 allele, after the first meiosis, we will have gametes that contains both alleles in the same gamete. So you see here, we have this gamete that has both the A1 and both the A2, while this gamete has both the B1 and the B2. And so these 2 gametes are candidate gametes for double reduction events at the end of meiosis second. So at this point, these gametes needs to go through meiosis second, but let me just focus about one case, this specific gamete. So this gamete needs to go through from telophase first to telophase second. And again, it can go through an equational separation or a reductional separation. Let's just focus on the reductional separation. So you can see here again, the alleles from the same chromosome are inherited in the same gamete in both cases. And you can see that the genome composition of the gamete comes just from a single subgenome, an A genome, where we will expect to have an A and a B subgenome. So these are 2 gametes outcomes of double reduction. And so when we cross this gamete, as I said before, with a regular Arachis hypogaea gamete with an A and B subgenome, you will have unbalanced genomic composition and increased diversity. And so how we estimated this process in peanut, you can understand that estimating this process is pretty complex, but we were able to do that thanks to a specific population structure, which is a BC1 population. And so we use as recurrent parent a regular peanut plant with an AABB genome composition, and as donor plant an induced allotetraploid peanut, which was created from the cross of Arachis magna and Arachis tenosperma. And so if we are looking at these BC1 progenies, we will notice that their genomic composition is composed by 1/4 of Arachis tenosperma. And this is really important because if we are using one-to-one segregating markers, we were able to identify the unbalanced genomic composition. And so we genotype this population with 384 SNP markers, and then we select them for polymorphism for Arachis tenosperma. So if we are looking at one specific marker with our genome software, we will notice the unbalanced genomic composition. So here on the x-axis, you can see the fluorescence intensity for the C allele. In the y-axis, the fluorescence intensity for the G allele. And you can see that each one of these dots is one of the BC1 progenies. And you can tell that the markers is polymorphic because you can see that Arachis tenosperma is here alone. And you can see that he has a G, it is homozygote for the G allele, and it has just the A genome. So being a BC1 population, what we will expect is that we have the homozygote for the opposite allele to Arachis tenosperma, which are these progenies in red, and the heterozygote, which are these progenies in yellow. But what is this? What is this cluster? This cluster can just come out from double reduction events in the induced allotetraploid in the male parent. And in fact, you can see that he has an AAAB genome composition, and it has a GGGC allelic combination, a novel allele combination. And so using these markers, we were able to create a 10-group linkage map. And here in red, you can see the hotspot for double reduction events. And so 21% of the progeny show at least one clear case of double reduction, and all the events occurring in chromosome 4 and 6. And what is really cool is that at the bottom of chromosome 6 here, there is a region responsible for seed size in peanut. And so when we have these cell outcomes of double reduction and they go through further meiosis, we will have more and more recombination and unbalanced composition. And so really the double reduction is acting as a genetic diversity enzyme for peanut crop. And so in summary, this is the first study that detected double reduction genetic phenomenon in a segmental allotetraploid peanut. And to our knowledge, is the first study that detects double reduction in allopolyploid species. And 21% of the progeny show a clear case of double reduction in the male parent, and all the events occurred in chromosome 4 and 6. And so the future the objectives of this project is to reanalyze this population. We have 48K SNP chip. And so in this way, we will be able to have a higher density genetic map and found other hotspots for double reduction and maybe infer other genetic phenomena. With this, I want to thank my lab, the Wild Peanut Lab, my advisor, and Mars to fund this project. And I want to thank you, everybody, for your attention.
Thank you, Samuele. Very good presentation. I apologize again for the mistake in the order. We have time for questions. Are there questions for Samuele? Any questions? We have a quiet audience today. All right, with that, thank you very much for presenting, and thanks for being a part of this graduate student session. We're grateful for that.